lab number 4 introduces some new material to us that we haven't really seen before. So I wanted to introduce the background concepts for this lab. Once I do that, you should watch the video that is posted on Blackboard about how to load a gel. As usual, carefully read your introduction and all of your exercise information before you get started. And as always, let me know if you have any questions. I would actually strongly suggest that you read through the lab and do the pre-lab first before you watch this video if you haven't done so, just so you have kind of an idea in your head of what I'm talking about as I go through all of these new words. So this lab looks at the relationship between DNA and protein. So of course we learned this in Chapter 6. In Chapter 6, we learned all about gene expression, which is the process by which you go from information encoded in your DNA or in your genes to a functional protein. Now, proteins are important because they are the molecules in our cells that do all of the jobs. They do everything from being the pigment, the color in your eyes, to making the pigment and the color in your eyes. They do things like produce enzymes, which are proteins that do work, that perform chemical reactions, such as the enzyme lactase, which is the enzyme that breaks down sugars in milk. So if you have the ability to do that, you can consume dairy products. If you are lactose intolerant, that means that your body does not produce that enzyme. So proteins do a whole variety of things within our cells. And we learned about this process of gene expression in 2 steps, transcription and translation. Now remember that you go from DNA to producing a short copy, a messenger RNA of the gene that you want to express, and then that messenger RNA is to produce a polypeptide or amino acid chain. And then of course we didn't talk about it too much, but that polypeptide is what is processed and turned into a functional protein. So before we start talking about proteins and looking at all of these things in our lab today, I want to introduce a few new terms to you. So the first one is a chromosome. So a chromosome is basically just a piece of DNA. So we learned about these a little bit in Chapter 4. Even though you didn't know you were necessarily learning about chromosomes, we learned that in bacterial cells we have singular circular chromosomes, and in eukaryotic cells we have multiple copies of linear chromosomes. In humans we have 46 chromosomes, and that 46 is actually made up of 23 types of chromosomes, and we have 2 copies each. So you get one of those copies from mom and one of those copies from dad. So you are probably familiar with a chromosome. If you've seen a picture of a chromosome, you often see them drawn like this. This is a sort of X shape. This is actually 2 identical copies of the same chromosome. This image here that you've likely seen if you've taken other biology classes are called homologous chromosomes. Sorry, that is not true. They are called sister chromatids. Okay, so these are actually 2 identical copies, kind of like photocopies that are still stapled together, or photocopies that are stapled together. And then you might have a second copy, or you will have a second copy. One of those copies comes from dad, one of those copies comes from mom. So we are going to actually look at this slightly differently today. We're going to remove those sister chromatids, and we're just going to look at the individual chromosomes that came from mom and from dad. Sorry about that. I thought I had that paused. Clearly I did not. All right, so we are going to look at these chromosomes, one that came from dad and one that came from mom. Now, we'll get into details a little bit later about chromosomes. If you don't fully understand that homologous chromosome and sister chromatid, the differences, don't worry about it. We will come back to that. But I did want to show you the difference just because sometimes students are like, oh, but they're X-shaped. They are, sort of. So anyway, let's move on, because what we really want to do is get from information in our chromosomes out to a protein, and that's what we are going to be looking at today. So chromosomes are pieces of DNA. We get one from mom and one from dad. We have 23 types, so you got 23 chromosomes from mom and 23 from dad to make 46 altogether. Now, encoded on these chromosomes are genes. So genes are specific regions that encode a particular type of information. Now, in this case, we're going to say that this is the gene for hemoglobin. So hemoglobin is the molecule in our blood that binds oxygen. So the purpose of our blood is to move oxygen from our lungs out to our other muscles and tissues that need to use that oxygen. So normal hemoglobin produces a lovely, or allows for our blood cells to be that nice, beautiful, round shape. It almost kind of looks like a donut without a hole in the middle. Now, if our hemoglobin gene has a mutation, it produces misshapen hemoglobin, and we get a disease called sickle cell. Now, many of you are probably familiar with this disease. In sickle cell, it causes a misshapen hemoglobin molecule. And when those hemoglobin molecules that are found inside your red blood cells are not formed correctly, it causes the red blood cell to take a sickle shape, this sort of crescent moon shape. Now, this causes all sorts of problems for the individual. One of the most important is that the blood cannot efficiently carry oxygen in its sickle form. And so there are a lot of issues with lack of oxygen to tissues. And then, of course, because these normal blood cells are nice and round, they usually flow through our arteries and capillaries and everything very nicely. But these sickle cell red blood cells get stuck because they can't flow nicely because of their shape, and so they cause a lot of pain and problems with different organs. So let's say that an individual actually has a gene that contains the mutation for sickle cell. Now, it is said that this individual has a mutant allele. An allele is a version of a gene. So here we have two different alleles. We have the normal allele for hemoglobin, and we have the mutant allele for sickle cell. So you can see that individuals can either have two normal copies. That was the first case that I drew with the normal hemoglobin. They can have one copy each of the normal and mutant hemoglobin, or they can also have two copies of the defective, the sickle cell hemoglobin. Individuals who have two copies of the same allele are called homozygous. Homozygous individuals have two copies of the same allele. Individuals who have two different copies are called heterozygous. Two different copies of alleles. So two different alleles. So they have a normal and a sickle cell. I don't know why it won't stay. I don't know why it won't stay blue there. All right. So we have our gene. Our gene refers to the region that codes for a particular trait. So in this case, this gene codes for something about our hemoglobin, whether it is normal or mutant. The alleles talk about which version of that gene that is. So one might be normal, the hemoglobin that produces that round blood cell, or it might be mutant, that hemoglobin that produces that sickle cell. A genotype is the genetic makeup of an individual. Now, the alleles are often given particular notations, so we know which one is which. So, for example, for sickle cell, the genotype of the normal hemoglobin is written as capital H. So that would tell us, if somebody has a capital H allele, that would tell us something about one of their normal alleles. The mutant, or the sickle cell, is denoted by an H prime. Notice I put a little apostrophe up there. So an individual might be homozygous for the normal. They might be heterozygous, one copy of each type, or they might be homozygous for sickle cell, two copies of the sickle cell allele. That is their genotype, their genetic makeup. So it's basically the notation for the alleles that we're talking about. Related to the genotype is the phenotype. So the phenotype is the physical trait that you see. So in this case, if we were to look at the blood of the individual, they would have the normal phenotype, normal hemoglobin, or they would have sickle cell hemoglobin. And something that's unique about the sickle cell is that if you are normal, if you have normal, if you are homozygous normal, all of your blood cells are going to be normal. If you are homozygous sickle cell, all of your blood cells are going to be sickle shaped. But if you are heterozygous for sickle cell, you are going to have half normal, half sickled hemoglobin. So half sickle blood cells. So let's talk about how this half normal, half sickled hemoglobin relates to the process of gene expression. So in our DNA, we might have a normal hemoglobin allele and a sickle hemoglobin allele. In the normal hemoglobin, we produce a messenger RNA, and that produces a normal hemoglobin protein. So I skipped some steps, right? After messenger RNA, we actually go through translation and we produce our amino acid chain or our polypeptide. And from there, we produce that hemoglobin protein. If we start with our sickled hemoglobin, we still produce a messenger RNA, but that messenger RNA is going to be a little bit different. It's going to have a mutation or a change in the DNA. We'll look at that mutation later in this lab. We'll see what actually happens to go from normal to sickle cell. Now that mutation is going to then produce an abnormal hemoglobin protein. So we are going to get a different protein because our mRNA was different, because our DNA has that sickle cell mutation. So a change in your DNA is going to lead, a change in your genotype is going to lead to a change in your phenotype. All right, so as I said, there's a video on gel electrophoresis that explains how gel electrophoresis works. So I'm actually going to skip over that in this video and go directly to what you are going to see at the end of your gel electrophoresis. So gel electrophoresis is actually a way for us to visualize DNA, or in this case, proteins. So we are going to actually have a gel similar to a really thick jello, and we're going to load our DNA or our protein into this gel and then apply an electrical current, and that's going to pull our sample through the gel. Now, as I said, you need to watch the video that goes with this that explains a little bit, and your lab explains a little bit more of the technical details of this. But I wanted to show you, and this picture comes from your lab, we can actually see our phenotype here. All right, so one of these, if you look here, we've got our alleles. Remember our notations for the alleles that we have in our DNA. Now these dots represent the proteins, excuse me, some technical difficulties here, some proteins that are extracted from a blood sample. So if you look at this individual, we have the homozygous mutant or the sickle cell alleles, and we have the corresponding protein for the sickle cell. Now, we have two alleles, but we have only one protein. That's because each allele is directing the production of one of the same protein, and so it all runs together sort of in a big blob. For the individual that is homozygous normal, we have the same situation. We have this purple protein, stained purple so you can see the difference, the purple protein, and we have two alleles, but we have only one type of protein because they're each directing the same protein. Over here, we have the normal, excuse me, the normal and the sickle cell protein being produced at the same time because this individual is heterozygous. So this individual is the heterozygous, and they produce both the normal and the sickle cell at the same time. So because each chromosome, each allele is producing a different type of protein, we actually see both of them. So you can use the pattern produced on the gel. That will tell you the genotype of the individual. That will tell you the phenotype. Because remember, the phenotype is the physical expression, what is actually being produced, which will then allow you to deduce the genotype. So if we were to look at a gel without them being labeled, if you see a blue, that indicates the presence of sickle cell. If you see purple, it indicates the presence of the normal hemoglobin. If you see both, it means that that individual is heterozygous. So now the last thing that we're going to ask you to do is to calculate the genotype or phenotype frequency. Okay, so you are going to have to determine if you are being given the genotype or the phenotype. And then you're going to have to figure out the frequencies of the individuals. So in exercises 4.1 and 4.2, you're going to look at gels and you're going to determine the genotype or the phenotype. So there is more information in your lab manual, but in general, to determine a frequency, you're going to take the number of events divided by the total number. So let's say we have 25 individuals and 15 are normal. So the phenotype frequency would be 15 out of 25. So 15 divided by 25 is 0.6. Okay, so that tells us that 60% of individuals are normal. Now, what if we wanted to find the genotype frequency? Okay, so this is a little bit different and this confuses students sometimes. Let's say we want to find the genotype frequency. Now we have to know something about the alleles. But let's say that 15 people are normal, 5 are heterozygous, and 5 have sickle cell. All right. Now, genotype, remember, that is information about our alleles. And remember that each individual has 2 alleles. So if they are normal, they are going to have, if there are 15 normal people times 2 alleles each, that means there are 30 alleles in our population, 30 normal alleles. Individuals who are sickle cell have 2 copies of the mutant allele. So 5 individuals with sickle cell times 2 alleles each. That means we have 10 sickle cell alleles. Okay, stay with me here. All right, so individuals who are heterozygous have 1 copy of each type. So those individuals have 5 normal alleles altogether and 5 sickle cell altogether. So let's calculate the frequency of sickle cell allele. Remember, frequency is the number of events that you're interested in divided by the total. So let's count up our total sickle cell alleles. We've got 5 sickle cell here and 10 here. So 10 sickle cell divided by the total number of alleles. Now remember, we have 25 individuals, each with 2 alleles, which means they have 50 alleles altogether. So 50 total alleles. So the frequency of our sickle cell allele is going to be 10 divided by 50. I don't know why I needed to do that. 0.2. That means that 20% of the alleles are sickle cell. So the last part of our lab is looking at how changes in the DNA affect the amino acid sequence. So we are going to effectively do transcription translation on paper by using complementary base pairing and then our codon chart to determine the amino acids. So I'm going to do the first one here for you, just so you get a little refresher on how to do transcription and translation. So recall that C pairs with G and A pairs with T, except in mRNA, A pairs with U. So let's do the first triplet here, our normal DNA sequence. If we have a C in our DNA and we make our messenger RNA copy in transcription, we are going to put a G in our messenger RNA. If we have an A in our normal DNA sequence, we're going to add a U in our messenger RNA. If we have a G, we're going to add a C. I'm just going to actually do the second one as well. So then if we have a G, we put in a C. If we have a T, remember the T's complement is A, A's complement is U. So now, I'm not going to finish this mRNA sequence, I'll let you do that. Let's go look at the codon chart earlier in the lab to determine the first 2 amino acids in the sequence. So let's remember GUC, CAU. So if our first 2 codons in our mRNA are GUC and CAU, that means that our amino acid is going to be GU, so GUC. So valine is our first amino acid. CAU, second is histidine. So you all will finish your mRNA sequence, finish your amino acid sequence, and then do the same thing for your sickle cell sequence. And then the lab asks you a couple questions about that sequence. What changes happened and what effect did it have on the amino acid sequence? So that's all that I'm going to go over for now. Please read your lab carefully. Please watch the video on gel electrophoresis. All of the data is available for you on Blackboard. And as usual, you are welcome to email me with any questions you have.